Hello, 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 and welcome to Austria's first ever Pint of Science Festival. My name is Harry, and I will be your host, MC, Master of Ceremonies, or whatever you'd like to call me this evening. I'm a master's student in the Department of Science and Technology Studies at the University of Vienna, and in my free time, I also try my hand at podcasting with the Science Slot Machine podcast, which I do with several of my colleagues. You can find that on Spotify if you'd like. But that's really enough about me, because tonight is not about me. I want to sincerely thank all of you for coming here and tuning in virtually for the second day of the Pint of Science Festival. We have such a fun evening planned for all of you with two amazing speakers lined up. And while we would love to be at the pub with you, especially with Vienna's bars and restaurants opening back up this week, we invite you to pour yourself a drink, whether it be a beer, a spritzer like me, or even a coffee or a tea, and make yourself comfortable wherever you are and wherever you'd like in the comfort of your own home. So go ahead, kick your feet up. You deserve it. And wait 1 second. Cheers, everyone. Now, during the event and this whole evening, our whole mode of communication will be the YouTube live chat, which you can find somewhere down there or on the right. That's how you can really easily use and participate the entire evening with your questions and comments. So to start that out, I would like to know, where are you all tuning in from? Go ahead and write that in the chat, whether it be here in Vienna, somewhere else in Austria, or anywhere else in the world. We'd really love to know where you're from. So before we get started with everything and I invite our two speakers, I'd like to first share a short video about the Pint of Science Festival in Austria from our director, Lisa Resnick.
Heute ist es soweit. Los geht's mit Pint of Science. Das Ziel von Pint of Science ist, Wissenschaft wieder stärker in der Gesellschaft sichtbar zu machen. Wir wollen die Wissenschaft aus ihrem Elfenbeinturm herausholen und die Bedeutung von Forschung für unsere Gesellschaft hervorheben. Dazu bringen wir 3 Abende lang die Forscher und Forscherinnen der Stadt in die Lokale nebenan. Dort präsentieren sie im gemütlichen Rahmen neueste wissenschaftliche Erkenntnisse und diskutieren deren gesellschaftliche Dimensionen. Unser Publikum braucht dafür kein Fachwissen. Die Vorträge sind an die allgemeine Öffentlichkeit gerichtet und bieten allen die Möglichkeit, mit Wissenschaft in Kontakt zu kommen. So wollen wir gemeinsam die Wissenschaft feiern und wieder für alle greifbar machen. Und natürlich soll das alles auch zeigen, wie viel Spaß wir an der Wissenschaft haben. Das Festival ist 2013 in England gegründet worden und hat sich seitdem in der ganzen Welt ausgebreitet. Inzwischen sind schon 30 Länder dabei. 2019 habe ich die Idee des Festivals auch nach Wien gebracht und von hier weiter in Österreich verbreitet. Dieses Jahr findet es zum ersten Mal statt mit Freiwilligen und Vortragenden aus Wien, Graz und Linz. Und ich hoffe, nächstes Jahr werden es noch mehr Städte sein. Ich wünsche euch viel Spaß bei unserem Festival und freue mich auf eure Teilnahme. Prost!
Okay. Hello again, and it's so nice to see that so many of you are able to join our virtual pub wherever you may be. I see many of you in Austria, but I also see many of you are not in Austria. I myself am in Vienna, and today I really tried to enjoy the few rays of sunshine poking through my window. As you might know, if you're here in Vienna, it didn't work out so well. But the city is opening up as we all try to figure out how best to live in life after Corona. Restaurants, pubs, movie theaters, and concert venues will soon open their doors once again, and I really couldn't be more excited. So like I said, we have a really nice evening planned tonight as we explore the topic of the beautiful mind. For about the next hour, our two speakers and I will guide you through this event, aptly titled Connecting with the Brain. During the event, while our two speakers give their talks, we would love for you to get involved with our discussions, again, by using the YouTube live chat somewhere around here. Following the presentations from our speakers, we are super excited to get right into your questions. If you're also on social media, please feel free to find us on Twitter, Facebook, and Instagram at Pint of Science AT, and please use the hashtag Pint21 to discuss your experience. Now, the brain. it needs no introduction, right? For most of us, it's just a mysterious entity we know very little about. Sure, we have some idea what it looks like, as you might see by our logo here. Yet, despite how modern the world around us may seem, there is simply so much we do not know about the all-controlling organ found in all of our bodies, the brain. It also seems to work in mysterious ways, or so we think. But how do researchers today study the brain? I mean, it's really not the easiest organ to get to, right? And while brains are certainly donated for study after death, these come with their own limitations. Motivated by the countless remaining questions for all things brain, researchers today benefit in many ways from emerging technologies and the new methods and techniques that these facilitate. These new ways of doing things enable new approaches and ultimately provide our societies with invaluable new insights into how brains function and, of course, dysfunction. Speaking of new and novel approaches, I would now like to welcome our first speaker, Guillaume Bellec of the Graz University of Technology. Hi, Guillaume.
Hi. Thank you very
Guillaume,
much.
of course, Guillaume. Nice to see you, man. Cheers.
Yeah, cheers. So I'm sorry, as I told you before in backstage, we didn't have beer at home. So that's just going to be water today.
Guillaume is pint of water game.
Yeah, but anyway, the science is going to be here, so we'll do with that, I guess.
Awesome. So Guillaume is a postdoctoral researcher whose research uses mathematical principles to ask the fundamentally challenging question, how does the brain learn? Although he is admittedly not a conventional neuroscientist himself, he does this by using his background in artificial intelligence and machine learning, which I assure you is quite extensive. Guillaume even co-developed an app that can help you learn to play guitar by detecting which chords are played and how they are played on the guitar neck. But today, Guillaume is not going to talk to you about his app. Maybe he might mention it, but what he's going to talk to you about is his latest research, which is closely related to an article he co-authored just last summer in the journal Nature Communications. Okay, Guillaume, I now cede the stage to you.
Thank you very much. Okay, so thank you very much to all of you for joining. I'm really pleased to be here, and thank you for the team who organized the festival for inviting me. It's really a pleasure and an opportunity to me to present my research, and to a big crowd like that. It's really a chance. Thank you very much. And so let's just talk a little bit about me before, and I will start with that. So who am I? I'm basically born and raised around Paris, and I always was pretty good at mathematics, and I loved mathematics, and I spent a lot of time playing music. Okay, because of that, I was at some point a bit tired of just doing theory, and like kind of school math. So during my master, I went to London to do machine learning for music, and you will see why I'm mentioning this, because I really started to like it. And after 6 months in London, I went to Stockholm for a year and did an Erasmus exchange, where I again did machine learning for music, and I started to ask myself a question. But okay, like we think about music and how we perceive music, and how music makes us feel emotions and so on. But technically, how is the sound coming into our brain, and then suddenly some electricity goes from our ear to the first neurons and the second neuron and so on, and this transforms into some kind of emotion? So I got into this big mystery of perception and emotions and so on, and basically the brain, right? So that's how I started my journey, I'd say, in neuroscience. And then I went back to Paris, and I started to ask myself, okay, how can a geek do neuroscience? So luckily, I found a good master where I could do at the same time machine learning and computational neuroscience. So basically, how mathematics can be a modern neuroscience. And from there, I found a PhD position in Graz, where I was very happy to go and discover this beautiful city that has been hard to leave actually since then. And so I finished my PhD there, even though it's in computer science. Everyone around me in this lab were fascinated by the brain, and that was really the main question we asked ourselves there. And since then, I moved to Lausanne, and I still do the same thing, but I come back and forth to Graz, and I'm really around a lot. So as he mentioned, I wanted to tell you about my app, but Harry already told you. But this is the name of it, in case you're wondering. So this is also just to tell you that you never leave where you're from, and I keep on doing music, and as part of my free time, I had to geek out this application and to help other amateurs to play music. So yeah, check it out. It's called Coda if you want, but that's really not the topic today. So the topic today is the brain, okay? And specifically, what I want to talk to you about is how do we learn to do things, okay? So some of you probably play ping pong, some of you can probably drive, right? And these are things that we had to train by, learning, doing it many times, and the more we do it, the better we do it, okay? But what is actually happening in our brain when this happens? So if I tell you where this kind of science started in this topic, it's in 1949, right? Where Donald Hebb, a psychologist, made this crazy hypothesis that basically everything we learn is maybe just explainable by a simple rule, which has been later summarized as this: neurons that fire together, wire together, right? So the idea is, as we learn to play ping pong or to associate things together, the idea that they're just neurons that pops up like that, they send electricity to each other in our brain, and the more neurons will fire at the same time, maybe the more connections between them will get stronger, okay? And he made this crazy postulate that maybe a simple rule like that explains, the whole way we learn, okay? And that was quite crazy, because at the time, nobody had actually opened the black box, right? It was impossible almost to look into a leading brain and record from it, and, in particular, through a learning experiment that requires, many to redo the same thing many times and so on. So it was a bit crazy to postulate something like that. It sounded like you cannot check that. You cannot verify this theory, right? But, why not? So just to summarize, maybe with a little bit of picture. So imagine the brain, right, in this big ball with a lot of neurons connected to each other, and we're gonna have this neuron I and neuron J. I will keep these names for these two neurons, which will be my two protagonists, right? And as I told you, every time they're active together, so this guy is active and then this guy is active, or the other way around, then maybe this connection here will get stronger. So maybe it will even grow a new dendrite or something like that, right? And the crazy thing is that 20 years later, after basically he made this theory, people started to realize that was true. So this is Bliss and Lomo from Norway and England, who in 1933 made this crazy experiment. So I'm showing here to you a figure from that original paper in 1933. And you see here what they did. So this is a hippocampus, right? Which is a part in the middle of the brain. And they managed to put an electrode in some neuron here and another electrode into the fiber that would be into the fiber that comes out from this neuron here. But so this is the electrode that you see here in their graph. And they excite them. So they send an electricity here, and then they send an electricity there. And they try to basically verify the theory of Donald Hebb 20 years ago. And surprising or not, they found that it was pretty much true. So if they force the co-excitation of those two neurons, it will get stronger, basically. Okay. So super interesting. That was in 1973, but then a lot of things have happened until then, and people have started to refine these theory, right? So imagine these are my neurons, and you see here on that direction would be time, and this bar says that at that particular time there is an electrical pulse that is coming out from that neuron, right? And here is some electrical pulse that is coming out from the other neuron, right? So as it's drawn here, so if this is a neuron drawn, they would typically do that by putting an electrode inside. And basically, they just realized that it's not just those two neurons that are important, but they also realized that sometimes there is this learning signal that is this dopamine. So you may have heard about this magic dopamine, which is chemical that has made the press because it's all over the place in the brain, and it's the chemical that you receive once you get a reward, right? So once you say expected something very much, and then suddenly it happens to you, and this is what is somehow flushed in your brain by a tiny population of neurons that send out everywhere. And what they realized here in particular is that this was crucial also for learning, right? So if that is flushed at the right time, then the neurons that were coactive will even grow bigger connection between them, okay? So basically, start to refine this experiment, and so that it was maybe a little bit complicated, and so that even there is this temporal delay potentially between the activity of that neurons here and really the moment when that dopamine arrives, but that together, by combining this with some kind of timing, coherent timing, you can perhaps explain how the brains learn, right? So this is kind of the stage of the experimental science on that at the moment, right? And then, so I will use the same terminology. So hegemoity trace just means there has to be something, you know, inside the neurons or inside the synapse, that when there is activity here in the neuron before the connection and the neuron after the connection, there has to be some molecular mechanism inside that retains some memory of that for a couple of seconds until the dopamine arrives, right? So because this is what it was shown experimentally to matter. So basically, we just call these things that we don't really know nothing about inside as eligibility traces. We just call them this way, okay? And then the other terminology that you are already starting to mention, that I will use the learning signal just to talk about these things like dopamine that are flushed at the moment you get a reward, to basically engage into the actual learning and make the connections get stronger. Okay. So how does that then, how can we model that? Okay, because my point here, I told you, is to talk about mathematics and show how the mathematical rules can maybe explain also how this learning is taking place and inform us more about that. So let's take the following task, where you have to imagine that a mouse will learn to do these things, and it's not quite easy. So it has to go through this corridor, right? And count those cues that you had at the beginning, right? So here it was 3 cues on the left and then 4 cues on the right. And then it has to memorize it during a delay. And at the end of this corridor, it has to go left or right, depending on which side had the most cues. So here it was 4 on the right side, which is more than the left side, so it has to go right, okay? And the mouse, so it's actually a typical experiment that people do in the lab to study learning and working memory, right? So they would put a mouse in this and then train it for a couple of weeks, and then perhaps open the brain and record to see what's happening, right? So this is what people manage to do right now, okay? And so the theory that I want to explain here is basically how somehow the neurons that are involved in this decision, right? When the mouse doesn't yet know what is the rules of the game, doesn't yet know if it's right or left and the cues that matter, right? How just the neuron that we fire for going right or left, suddenly the moment the mouse identify that, oh, that was right, this is where I got the cheese, this is where I got the rewards, but this will actually somehow induce a learning about what has happened in the recent past, about what is in the working memory of the animal, to say, okay, how, I mean, it's not necessarily a conscious process, but how do these things have to organize to basically understand the rules of the game and learn to solve this little puzzle properly, okay? So how do we model that mathematically? Okay, so we will consider that each neuron is this thing here that we simplify a lot. We don't get in the full complexity of what it is because we want to do mathematics, okay? So we will consider that all the dendrites here are just summarized by all the electrical inputs that comes from all the neuron, which are those numbers. They're just weighted by a number, right? So if this is a strong connection, it will have a strong weight W, otherwise it has a low small w, okay, if the connection is not strong. And then these neurons, so everything that happens inside here, we just put, we just sum all the inputs and apply a simple mathematical function to this, okay? So it's a very simple, simplified model of the neuron. So if you want to see dynamically a little bit what it would do, okay, so it would look like that, okay? So all the inputs are coming in, right? So this neuron here would be an input, and everything that you see here is the neuron J, which would be the neuron I'm talking about. So you see here inside in yellow, it will integrate those inputs, and then suddenly if the voltage, this yellow voltage here is high enough, it will cross this purple line, and then the neuron becomes active, and then it would send, therefore, activity to the next neurons. Okay, so this is really a very simplified model of a neuron, but then the idea that now, because it's so simple, we can do mathematics about it, and we can make a model of many of them. Okay, so, and then when we make a model, we want, as I told you, to see how learning is happening. So from that, we will take hindsights from artificial intelligence. Okay, so what I mean by this is that you probably have heard about those results from the research team at Google, who managed to play chess and Go and other games with basically artificial neural networks, right? So these models are, in fact, not so different from the model I just explained to you, right? It's just bigger, and it's perhaps not so much meant to resemble biology, but it's just meant to play chess, right? So they maybe simplify it even more, but basically it works similarly, okay? And then how do they learn to play chess, right? They do something which is called gradient descent, which is just basically finding a rule to change all these blue synapses here. So as I told you, all these connections between two neurons, they will train them to make their algorithm better at playing chess and Go, right? So how does that work? Is that, say you start here with a naive network that doesn't really work, then you formalize mathematically that you have an error function or loss function, large E here, which is represented in this nice curve here. And this is basically how much error you've made, how much mistake you've made during the game. And your goal is just to reduce this as much as possible. And then this gradient descent algorithm is just telling you, just like a skier would go down a slope, it's telling you, okay, so if you want to go down like this, you have to change this synapse by that much. Or if you want to go down like this, you have to change this connection by that much, and etc., right? So by doing this, it's changing the connection of the algorithm until you go down, all the way down in this error function, until the bottom where the algorithm can really play well at chess or Go. Okay, so how do we take that and bring that to brain research? Okay, so the problem is, again, the idea there was just to train an efficient algorithm. So if we model how a mouse doing this exercise would learn with that system, okay, so we model the brain, as I told you, those are some neurons, so they're just exchanging spikes, how the slope reset is going. And then what the learning algorithm would do, it would go backward in time, right? Which is time travel. It's completely impossible. You can't imagine that the real brain can just go backward in time, right? So why does the algorithm go backward in time? Because it has to find out which connection between two neurons needs to be changed for the error to decrease, for the number of mistakes to decrease, right? This is called the credit assignment problem. And in the brain, you can't go backward in time. So how do you have to make up for that and find an alternative algorithm? So this is what we basically discovered there. Oh, Zos, I'm sorry about that. Let's look at this nice second video. And the idea is that as the mouse goes through the maze, right, instead of having to go backward in time, we have, just like I showed you, just like what was found experimentally, we have these blue things here and these red in these green things here, which are basically eligibility traces. So what I told you at the beginning, what it is just some mechanism that memorize within the connection what was happening locally between the two neurons, and then at the end, but at the end when the decision is made and then the cheese is found, those little trace of what has happened will be combined with the dopamine to make learning happening. Okay, so this is basically the theory. And by doing that, we manage to basically mathematically redo the exact same thing at what they do with the machines, but this is something that is possible and plausible for the mouse brain or the human brain, right? So it's a completely different theory and rather and then applicable. Okay, so this is an ugly formula. So this is a formula that basically summarizes when the mathematical theory that we derived. So you don't have to understand it. I will just go through a little bit so that you have to understand the components. So the first one here are the eligible traces I told you, right? So this is just when two neurons are active, locally within the connection, there is some molecule that just moves around and somehow memorizes that the neuron were active or not, okay? And then there is a learning signal. So this is not quite yet the dopamine, but this is, say, a third neuron, right? That when it fires, send a signal to the neuron J here, and that is actually one of the things we found out to be important for the learning. Okay, and then there is the thing stays around for a little while until perhaps it has happened that the mouse found the cheese and get the reward, therefore get the dopamine, and this is finally the moment where all that is combined into something that makes a change in the connection that will last for, say, minutes or days, depending on the situation, okay? So basically, this is now not necessarily what is seen in biology, but this is mathematically what is needed to make a system, to make a model of the brain learned just as well as machines from Google to behave in an environment, okay? And putting all that together, just to show you how what it can do, we played here Pong. Okay, so this is a virtual, say, the virtual version of the ping pong, right? And on the green here, we have a player that we actually trained with our plasticity rule, right? With our model of biological learning, right? On the left here, you have an agent that was hard-coded into the video game. So it's not something that's learning or anything, right? And the red player here, as you see, it's winning quite a bit. And for doing that, it had to go through hundreds and hundreds of learning trials, actually hundreds of thousands of learning trials. So you see, it's learning a little bit slow, but it's learning to win. And it's learning with a learning model that is biologically plausible, right? And what is important in this is that the theory of Hebb, as simple as it was, would not work for something like that because it was too simplified, right? And it didn't get into the complexity not only of the brain, but also the complexity of the learning mechanism that have to be involved. So this basically algorithm here, it's the first biologically plausible algorithm that was able to learn such complicated things with mechanisms that are supported biologically, okay? And if you want to see basically all the variables that are involved, they're here. And this is just to show you that everything that is simulated here in the agent are actually just the things that I told you about. So the spikes, the activity, the pulsative activity that exchange between the neurons, the eligibility traces, and so on. Okay. So in summary, okay, what I told you, we had a theory, we built a theory for how learning happens in the brain, okay? And this learning is basically the idea that the connections between the neuron get stronger or weaker depending on the experience of the animal in the environment, okay? But mathematically, by deriving these exact rules that are important for making this change efficient, we could show that some simple rules like that are compatible with what is seen experimentally enable a machine to learn just like basically Google's machine learning algorithm, okay? And then, of course, this experiment, you might say, okay, but what does it tell us about the brain? So that's where we are now, but this theory actually has some details like this learning signal that I told you, or the exact timing of the AGB traces that were needed for this to learn, which are then prediction of what needs to be tested experimentally. So now in Lausanne and with other collaborators, we're trying to go in the wet lab. In the wet lab, it means look at what's actually happening in the animals and trying to see if we can actually measure what the theory is predicting. So far, I cannot tell you if the theory is right as it is, if we're going to have to change it, but hopefully in the next years, we'll have a better clue and I can tell you whether that's happening or not, right? So the following talk, as you will see from Catharina, it's not exactly this type of, she's an experimentalist, so she's the type of people I want to collaborate with, but she's not interested in this exact same question. She will show you something different, but at least you will see a little bit more about what are those wet lab that I'm talking about, and a bit more about the daily life that our colleagues experimentalists are going through, okay? So thank you very much for your attention. And thank you very much for staying until now. I hope I didn't bore you. I hope my questions were not too scary. I guess it's up to Harry to come back and Catharina to come in for the next talk. And I'm looking forward to hear what she's going to present to us.
Guillaume, thank you, man. It was a super interesting presentation for me. And I can already see that there's a lot of comments coming in here on YouTube. So I really encourage the whole audience, if you can think of any questions that come up in the meantime, I'm going to shortly invite our next speaker here. But if you have any questions remaining for Guillaume, please post them in the chat there. So without further ado, I'm going to go ahead and introduce here our next speaker, which is Catarina Martins Costa of the Institute of Molecular Biotechnology here in Vienna. And like Guillaume, Catarina's research is quite groundbreaking and builds upon many recent scientific breakthroughs, especially though when it comes to using some very specifically treated stem cells to create what are essentially models of the brain, or as Catarina will tell you, organoids. The focus of Catarina's research is on the flow of information between the two different hemispheres of the brain. That's right, we have two, which execute completely different functions, but nonetheless rely on one another at every step of the way. Tonight, Catarina will talk to you about how she is able to do her research and what happens when these hemispheres do not exchange information correctly. So, Catarina, the stage is now yours.
Thank you so much, Harry, for this very nice introduction. You nailed the topic that I will be covering here today. Hello, everyone, and thank you for joining here today. I am super excited to be a part of the first ever edition of Pint of Science Austria. And today I would like to share with you a little bit of my research that I've been developing at the Vienna Biocenter in the context of my PhD. So, in general, I am interested in the human brain. But before starting this project, I had very little knowledge about the amazing complexity of our brains. But as Harry already mentioned, I also had this idea that the brain has two hemispheres that execute different functions. And one of them is more creative, whereas the other one is more analytical. And this is exactly what is here represented in the Pint of Science logo for the segment, Our Beautiful Mind. So this concept is not exactly true, but what is true is that the two hemispheres do execute some functions that are singular to them, meaning that some functions are lateralized. And let's look at a more specific and simple example for this. So something curious is that the visual information is processed in the opposing hemisphere to the respective visual field. So information that reaches the left visual field is processed in the left visual cortex, which is in the right hemisphere of the brain. On the other hand, the information from the right visual field is processed in the left hemisphere of the brain. So with this, the left visual cortex is, for example, very far away from the speech center, which is located in the opposing hemisphere. This means that when a certain visual information, for example, the image of a flower, is only seen by the left visual field, then the information needs to travel across brain hemispheres for us to be able to put into words what we saw. So in this case, to be able to say out loud that we saw a flower. But how does this happen exactly? So in our brains, we have millions of neurons. If we look at the brain as a map, every neuron can be seen as a city, and the cell body is where this city is located. But the neurons don't exist individually. So they are connected to thousands of other neurons, the same way that streets or bridges connect different cities. And these connections are made by structures known as axons. And there are short-range connections, such as small streets, but also long-range projections that can be seen as very long highways. And the region of the brain where these different long-range projections come together and converge is called the corpus callosum. So the corpus callosum is a structure composed of millions of axons that make the interhemispheric communication in our brains. And it is a very important structure. But one of the most common brain abnormalities is actually the absence of the corpus callosum, known in medicine as corpus callosum agenesis. And when this happens, if we look back at the previous example, someone that has a split brain will say that they saw nothing, as the visual information has no means to travel from the left visual cortex to the speech center to be put into words. So now let's look at some real brains. Here is an adult human brain so it is a healthy brain with the corpus callosum being this structure. And here is an example of a brain of a human with corpus callosum agenesis. So the two hemispheres are split and there is no corpus callosum. So this is a condition that is quite common, and it affects about 1 in 4,000 newborns. And the symptoms can be very variable. So there are people who are completely asymptomatic, but others that are severely affected by this condition. And most patients present intellectual disability, motor and speech delays, and are very commonly also in the autism spectrum. So as you can imagine, this disease can have a very big impact on patients and also their families. But many disease mechanisms and also the cause of the disease is in many cases not known. So there is an effort now in science for us to better understand the agenesis of the corpus callosum and to reveal new disease mechanisms. And the main model that is used to study the human brain is actually the mouse brain. And as you can see here, the mouse brain also has a corpus callosum, is this structure here. And it is also possible to generate mice with split brains, so with agenesis of the corpus callosum. However, if we compare the mouse and human brains, the differences are striking. The mouse brain is much smaller, and also it is completely smooth, whereas the human brain is very much larger and also has these convolutions. So due to these differences and many others, and also in an attempt to minimize animal experimentation, we are now coming up with new models to study the human brain. And I am using one of those models. So all my research in the lab starts with one single human cell. But this is not any cell. So it is called a pluripotent stem cell. And it has mainly two characteristics that make it very special. The first characteristic is that it has a virtual infinite capability of generating cells that are equal to itself. And to this ability we call self-renewal. And secondly, it also has the ability of differentiation, meaning that it can give rise to any other cell type in our bodies. And in the lab specifically, we can guide this differentiation to make the cell type that we want. So we can make heart cells, kidney, liver, intestine, or in my case, we can make brains. So these cells are really amazing, but one pitfall is that they do not exist in the human body. They only exist transiently during embryonic development, and then they disappear. But there was one Japanese scientist called Shinya Yamanaka that rather than seeing a problem in this, saw an opportunity. He thought that maybe it would be possible to start from adult cells and bring them back in time to when they were pluripotent. And Yamanaka and his colleagues were able to achieve this amazing goal and to generate induced pluripotent stem cells. This was such a revolutionary technology that nowadays there is a whole field of science that uses these cells to study different things. And as you might be guessing, I am included in this field of science. So all that we need to generate induced pluripotent stem cells from any of us is an initial collection of 10 milliliters of blood. From this blood, we then isolate cells with a single nucleus, and then we expose them to pluripotency factors, also known as Yamanaka factors. And after 2 or 3 months, we have induced pluripotent stem cells. So now I would like to show you actually how we maintain these cells and also how we use them. So let's do a short visit through the lab. Starting with the cells, they are usually kept in these plates with 6 wells, only this one has cells here, and they grow adhesive to the bottom of the well, and over them there is a medium with the necessary nutrients and growth factors to keep them alive and happy. And we can look at these cells with a transmitted light microscope, and here you will see them at a 5 times amplification. So we are peeking into the cells, and you can see that they grow in colonies. There are colonies that are smaller, whereas others are bigger. And basically, over time, these colonies grow, the cells replicate until they are occupying the majority of the well, which is the equivalent to about 2 or 3 million cells. And when this happens, then we take a portion of these cells, we put them in a new well, where they again form small colonies that grow, and the process repeats itself every 3 to 4 days. But now you might be wondering, okay, how does one use these cells to study the brain, right? So actually they are the starting material to produce what we call brain organites. And I also want to show you a video that is representing the process of generation of these brain organites. So the starting point is the cell colonies that we just saw. And then we make a single cell suspension, so individual cells out of these colonies. And we take about 9,000 cells that we place in a very small well with a U bottom. These cells are initially individual, but they rapidly aggregate and form a small sphere, which is the organoid. We keep changing the medium to provide the organoid with the growth factors that it needs to become, so to produce brain cells and also to grow. And after about 10 days, the organoid is ready to be transferred from this very small well into a petri dish. And in this petri dish, the organoids continue growing, and we also embed them in a matrix, a type of gelatin that gives it support for the growth. Initially, they are kept in suspension without agitation, but later on, we put them in a shaker where they continue growing for many days or many months. And in its final form, the organoids are 3-dimensional tissues composed of brain cells. So I also want to show you how this looks in reality. And these are the plates that we use to make the organoids, where they are kept in the first day. So these are the U-bottom plates, and the organoid is this small sphere here. And as you can see, we don't make one organoid at a time. So every one of these plates has 96 wells, and every well has one single organoid. And also at this stage, we can use a transmitted light microscope to check the organoids. And at this point, they are just small spheres with less than 1 millimeter in diameter. But they continue growing and maturing, and this is how they look after 100 days, these small white spheres. Even though they are already at day 100, they don't grow any more than this, but the cells that compose the organoid keep maturing and producing more advanced cells in the development of the organoid. So just so you also have an idea of the scale at which this is done, this is a shaker where I keep some of my organoids, and in each one of these shakers, we can keep about 1,000 organoids in these different plates. And in the lab, we have maybe 10 shakers, so 10,000 organoids growing at every moment. Between different cell lines and also different types of brain organoids, we also have to repeat each experiment at least 3 times to make sure that the results are reproducible. So everything needs to be done at a very big scale. Okay, but now that we already took a visit through the lab, let's go back to the initial question of the corpus callosum. As I told you, the main model used to study the corpus callosum is the mouse. And here we have a mouse brain where the corpus callosum is marked in white by the marker neuropilin-1. And the first part of my PhD was focused on the question of whether organoids are also able to generate such structures. And although I don't have a final answer to this question, I have several clues. So here is a human brain organoid at day 120 of development. And similarly to what we see in the brain, also in the organoids, we see the formation of axon bundles that are marked by neuropilin-1. And at this point, we are working under the hypothesis that these are the organoid equivalent to the corpus callosum that we have in the brain. So right now, we are working with a model that is in between a real human brain and a mouse brain. And as every model system, it comes with advantages and also disadvantages. On one hand, it is closer to the human brain because it uses human cells. But on the other hand, it is also further away because even though it is a 3-dimensional tissue, it is not a real brain. So the next step will be to see if we can use organoids to model corpus callosum agenesis. And for this, all we need is to derive induced pluripotent stem cells from patients that have this condition. So in the scope of my project, we established a collaboration with the Vienna General Hospital, the AKH, where they basically centralize all cases of brain abnormalities that are detected during pregnancy for the whole of Austria and also the south of Germany. And they have a vast cohort of patients with corpus callosum agenesis, and we analyzed the DNA of these patients, and we identified patients that have mutations that might be causing the agenesis of the corpus callosum. And within these patients that have these mutations, we were fortunate enough that 2 families agreed to collaborate with us and to be part of our study. So together with these families and also the doctors at AKH, we collected blood from these 2 patients and we generated induced pluripotent stem cells. And I am very excited that now we already are growing organoids from these cells and from these patients. And as well as the patients, both the cells and the organoids also harbor this mutation, which is here represented by the asterisk. However, we don't want to stop here. Every experiment that we do in the lab needs to have a proper control, meaning some condition that is not perturbed. In this case, we need to generate organoids where this mutation is absent. So to do so, we resort to a gene editing technique that is called CRISPR in order to modify these stem cells and replace the mutated part of the genome with a part of the DNA that does not have the mutation. And from these new cells, the control cells, we can again generate organoids. And then the question comes of what makes these organoids different. We know that at the level of the DNA, this mutation is the only difference. So we can basically establish a relationship of causality between any difference that we see in the organoids and the mutation that we identified. And although nowadays the organoids are mainly used to increase our knowledge, we are also working towards using them, for example, to test new drugs, or also to help us in efforts of personalized medicine. And of course, all of these small enigmas that I presented here to you today are inserted in a much larger question. And this question is, what happens in the development of our brain during gestation and how can something go wrong so that neurodevelopmental disorders arise. And of course, this is a very ambitious question, but also this is what makes it so exciting to work in this field. And I am still, after 3 years, marveled that this whole research can start from one single cell. So with this thought, I want to bring the presentation to an end. I want to thank all of those that have been involved in this project, our funding sources, also the amazing infrastructure that we have here at the Vienna Biocenter. I also want to thank the leader of the research group where I'm working, Jürgen Knoblich, and the amazing team of colleagues and friends that I work side by side with, especially Nina Corsini, who is supervising this project. A big thank you to the doctors at AKH with whom we are collaborating, and of course to the patients and their families. Again, thanks Pint of Science, and thank you all for listening. And now, cheers, ching ching, Proust, and we are ready for questions.
Catharina, Prost, cheers, and thanks for the really amazing presentation. And I'm so excited. We have so many questions, you know, that we really don't have so much time, but we're going to really try to get through as many as possible and take as many as we can. But first, as we enter our discussion round, and I talk very fast, I would like to invite everybody to maybe refill their drink. Mine's getting a little low here. And I would like to now invite Guillaume back to the discussion so we can enter our discussion round and take some questions. Hi, Guillaume. Welcome back.
Hi again.
So it looks like we already have our first question here. And this, I think, someone might have to correct me. I think this is a question directed for Guillaume, but someone might take it.
Yes, I think.
Siamalo wants to know, in a biological scenario, dopamine equals reward. Are there neurotransmitters associated with loss or failure? How is this replicated computationally?
Brilliant, brilliant questions. And the fact is, it is very complicated, and I don't really know the answer. I mean, I don't really know the answer. I've studied this a little bit. I was collaborating with neuroscientists who were looking at actually the dopaminergic neuron, but when the neuron is shocked with an electrical shock, right? So it's not a reward. It's more of a penalty, right? Or like even maybe a little bit of pain, right? And what happens is that quite some dopaminergic neuron were active in that moment, right? So I'm just saying that to say that even the simplification reward is dopamine is an oversimplification, right? And outside of that, there are many other neurotransmitters. So often when we would like to think of dopaminergic neuron as related to something as simple as this, so dopamine is surprise, or I don't know, so serotonin is love and things like that, it's often wrong. When you start to actually look at the data, this is an oversimplification of the reality, and often a single emotion is involved with a bouquet of neuromodulators that goes all over the place at different time with different timing, and the story is quite complicated. Could it be a good or bad emotion? One thing that's maybe good news is that people prefer in experiment to give rewards than to give penalties or shocks in general. So the good news is that for the animals, at least they get more cheeses, or actually it's not cheese, it's sugary water usually, but they get more of that than, you know, actually things that would reflect a failure or pain.
Guillaume, thanks for your answer. I will always remember when neurons fire, things get wired or something like that, but I will keep it simple for myself, but I really enjoyed your reply. Our next question here is from Irabar7, and they would like to know, they have a question regarding this split brain thing. So, Catharina, if someone got lobotomized, would this lead to the same symptoms?
So this is an interesting question. I think that so a lobotomy is different from a callosotomy. So the callosotomy is when you cut the corpus callosum. And this is usually, or often done when, for example, people have epilepsy, and the epilepsy has a focus on one hemisphere and then as travels to the other hemisphere, and there can be certain procedures that you cut this communication so that you also prevent the spread of the seizure. So regarding whether the symptoms are the same or not, so people so the corpus callosum, when it is cut afterwards, after it has been formed, they also experience, for example, some problems with lateralized movements, and also in this left visual field and speech center communication, they basically lose this ability. But what is very interesting is that when the patients are born without a corpus callosum, there can be cases where they don't have any symptoms. And this happens when there are other pathways that form and replace or kind of make up for the loss of the corpus callosum. So there is a range of symptoms that these patients can have. And when people are just the corpus callosum is cut, then they will have some symptoms. Yeah.
Super interesting. I do not want my corpus callosum removed, please, and thank you. The next question I have, this is for Guillaume. It's from Igor. He would like to know, what are the applied uses of this knowledge? Do you think we'll be able to improve the learning process somehow? Make it more efficient, perhaps?
Great questions. And I wish the science would be ready to already foresee this type of applications. But at the level where we are, understanding the good, say, rules that explain how each single connection changes would already be a fundamental progress and real plus, right? But understanding how one connection changes is very far from telling us how do we, on our daily life, can decide to do things differently to learn better, right? So for this type of understanding of the brain, I think the best type of research are typically cognitive science and so on, where you basically take somebody, do some exercise, and I don't know, take a nap or not take a nap, and then understand whether taking a nap is actually helping you to learn, right? So there's a lot of active research in this type of field, and the type of fundamental research that I'm doing at my level, I think are very hard with the current knowledge of the brain to connect to really the real use of, our real experience of when we learn things. But, this is fundamental research. In 20 years, who knows? In 40 years, who knows? I'm hoping for at least.
We have to. It seems that our next question here is from Melanie, and I think this is here for Catharina, using my lay knowledge here. And I think this says, how can some people with agenesis be asymptomatic?
So this comes a little bit into the direction that I was talking before, that so the brain is very plastic and it has an amazing capability to adapt. So especially not in an adult state, but during development. And what can happen is that when there is a problem and the corpus callosum cannot form, some alternative routes of communication can form. And these routes would not exist in the brain, in a normal brain or a healthy brain, but they kind of make up for the loss of the corpus callosum. And then the people that have a loss of the corpus callosum might not even know it, because they can still perform all the functions that anyone else can perform. So it's basically this plasticity of the brain and its ability to adapt to the loss of the corpus callosum that can result in asymptomatic patients.
Perfect. Thank you, Catharina. I think we have time here for one more question, and that is from Lone here. And this, it seems to be here for Guillaume, and that is keeping the analogy of with the mice learning, what cheese do you give to the computer to make it learn? Or can mice really count? This is what I get out of this question, but what do you give the computer to make it learn?
Yes. Well, so I want to cite here a very nice comment that I saw actually in reply to that in the in the comment thread. So somebody said a bite of cheese, and I think that was just a brilliant joke. So I just wanted to repeat that, bite, B-Y-T-E. Anyway, that's perhaps a geek joke, but I really laughed when I read it. And no, actually, it's a very good question. And yeah, exactly. And anyway, so so the answer is quite simple. Is in the Pong game that you saw, right? There is actually a score, which is just each time the opponent loses, doesn't catch the ball, it loses 1 point, right? And that is basically what we give to the learning agent. So each time it scores, we give a +1. And this is basically the unique reward that we give, and we think it's kind of at the same time a simple model of behaviorally relevant reward that is not too artificial and probably relevant to the happiness we get when we finally score in ping pong.
Mm-hmm. Well, Catharina, Guillaume, we're a little pressed for time here. But what I would invite our audience to do, if they want to get in touch and ask more questions about the magnificent research from Catarina and Guillaume, to please get in touch with them on Twitter. We will put their handles right here below for both Guillaume and Catarina. You can find them pretty simply using those handles. Thanks, Guillaume. Thanks, Catarina. Cheers, and thanks so much for joining us tonight. It was my pleasure.
Cheers.
Cheers.
Thank you very much.
Thank you so much.
All right, everybody. And now it's my turn to close it out. And so as it goes, any event like this features lots of thank yous. And so besides our speakers, I'd also like to extend thanks to our organizing committee. And maybe they can wave and say hello to everyone as I do that. So thanks to Katja, Anju, who was also the master of comments and really made my life a lot easier this evening, Mariella, Federica, and finally, our producer, Max, who took care of managing the very complex network of webcams and microphones for the entire night. So thank you guys all so much for your very hard work behind the scenes. And cheers. Cheers. Thanks, guys. So lastly, as we get out of here for tonight, thanks most of all to you guys, the audience. I know I had a lot of fun, and I hope you did too. Thank you for tuning in from everywhere. We could see that it was not just in Austria, but going far beyond and giving our speakers so many nice questions to answer. If you did enjoy this evening, I really encourage you to check out our remaining 2021 program, with events continuing on both tonight and tomorrow. And if you'd like to learn more about our events, please see the website below, pintofscience.at. Also consider there subscribing to our newsletter, which can be found right on the homepage. And well, that's really it for me and for tonight, guys. It was an absolute pleasure to be here with you all, even if only here through our screens. I hope to see you all at a Pint of Science event in person near future. I'm wishing you all a very pleasant and nice evening. Take care, and I hope to see you soon. Cheers. Thanks, everyone.